Hi everyone, my name is Joao Garcia. I'm a research fellow at the European Space Agency working on cultivated meat for human space exploration. First of all, I'd like to thank the organizing committee for the invitation to present some of the work we're doing at ESA on cultivated meat. So the current space food system is pretty much based on prepackaged food that is prepared on Earth, packaged and sent to the ISS. And is mainly composed by thermostabilized foods, followed by freeze-dried foods and irradiated foods, with a very limited amount of fresh food products. And on the ISS is resupplied quite often with these products, and therefore we also don't need to think about or to worry too much about the long-term stability of these products. However, if we think about long-term space missions and long-distance space missions, that will then expose the crews to extreme conditions, we know that adequate nutrition can help as a mitigation strategy for some flight-associated risks, such as bone and muscle loss. We also know that the current space food system might not be able to provide this adequate nutrition because we might not be able to resupply the crew so often. We also know that the shelf life of most food products is from 2 to 3 years, but already after 1 year of storage, we start seeing some degradation of certain nutrients. We also know that the crew might face some reduced food intake, and this is due to altered perception of taste and flavor, but also to the lack of freshness in food products. And we also have to consider that packaging accounts for 50% of the total weight of the food system currently. So it's worthwhile to think about and to research future and potential space food systems. These include, at the moment, plants and vegetables, microalgae and cyanobacteria, among others, and also cellular agriculture and cultivated meat, which is what I would like to talk about today. So cellular agriculture describes the process of using cell culture to produce commodities, and that can be meat, milk, or leather. And indeed, cultivated meat focuses on the meat as a product and describes the process of growing animal tissues such as muscle and fat using these cells. The core principles are derived from tissue engineering and regenerative medicine. It emerged as an alternative to the traditional and conventional livestock farming. It's thought to be safer because we would have complete control of the bioprocess, reducing the possibility of contaminations and therefore of foodborne illnesses. It is also thought to require less land and water when compared to livestock farming, and in theory could be performed with any species. These are some of the products that are currently in development in the cultivated meat space. So basically the way it works is that cells are collected from an animal, isolated and grown in bioreactors by giving them certain nutrients, oxygen and water. They're grown over a period of time and they're proliferated and then maturated into certain tissues and then harvested in an unstructured product such as a burger or minced meat, or a more structured product such as a steak or a fillet, and this is obtained by using scaffolds or 3D printing technologies. So of course, if you think about bringing these technologies from Earth to space, there's a couple of constraints that we have in space that we don't have on Earth. And I broke down these into 3 categories. So first, the space and habitat environment, you would have altered gravity, deep space radiation, and volume constraints. Regarding the system would likely need to be fully or almost fully automated and highly reliable and stable, since we don't have so many opportunities to repair, maintain, and even supply spare parts for the system. Regarding the bioprocess, the resources should be minimized, as well as the waste production. And we do have to come up with waste management and recycling strategies. And finally, the nutritional profile of the products should be stable throughout the mission duration. If you break down the system into inputs and outputs, as inputs you would have scaffolds, cells, media, power, and gases. As an output, the main output would be food, of course, but as waste, then we would have wastewater, lactate, and ammonia as the main byproducts of cell culture. Then you would have some desired capabilities, such as monitoring, automation, sterilization, recycling, and post-processing. And now I'm going to talk a bit some of these parameters. So regarding the cells, most of the cells being used currently for cultivated meat, or most of the species, are mammalian species, such as, or the main products are beef product and chicken, because pretty much most of the research is done on these cells. Although fish species might be more convenient for space applications because they're more resistant to variation in environmental conditions such as pH, because the fish species have naturally a higher buffering capacity, so they can resist to higher changes or more variation in the pH of the media, and they can also be cultured at lower temperatures, which might be an advantage in terms of energy demand. Regarding the cell type, I would mainly divide it into two cell types. So we have the muscle-resistant cells or progenitor cells. These are mainly the satellite cells and fibrodipogenic cells that are used to produce muscle and fat. These cells have limited proliferation capacity and long doubling times. However, they're easier to isolate, and the protocols are slightly easier as opposed to the pluripotent stem cells. On the contrary, pluripotent stem cells are a bit more difficult to reprogram or program. The differentiation protocols are slightly harder and a bit more complex, and these cells are also a bit more difficult to obtain, although they do have unlimited proliferation capacity and can be used to differentiate into two different or more than one tissue type, such as muscle, fat, and connective tissue. They also have short doubling times, which would make the bioprocess much faster. Regarding the bioreactors, there's essentially three types of bioreactors. So the stirred tank, which consists of a tank where the cells are cultured, either in single cell suspension, microcarriers, or cell aggregates. Inside the tank, there's an impeller that rotates the media and efficiently mixes cells and media. Then we would have packed bed, in which cells are cultured in microcarriers that are packed into a bed, and media is perfused through this bed and through the microcarriers. The main issue with this bioreactor is that it's very difficult to scale up, because as you increase the thickness or the length of the bed, you start having problems with mass transfer and you start creating gradients in nutrient and metabolites. Then you would have some, the holofiber would be the last one, and this bioreactor tries to mimic tissues. So you would have these very small fibers where the media is being perfused through, and these fibers have pores that allow the media to go through the interstitial space where the cells are located. So this tries to mimic pretty much the blood vessels or blood capillaries in the body. As opposed to the packed bed, the holofiber in theory wouldn't have so many problems in scaling up, because as you increase the length of the fiber, you wouldn't have so many problems with mass transfer or nutrient gradients. Regarding recycling strategies, so as I mentioned before, lactate and ammonia are the main byproducts of cell culture, and we know that ammonia can be metabolized by some bacteria and some microalgae. At ESA, there's a project being developed on a circular system composed by different compartments, so the crew, some bacteria, microalgae, and plants. So in this system, the crew provides the waste such as urine to some bacteria, which should then convert these wastes into nutrients given to microalgae or plants, and these products should be used as food for the crew. So you could think about a similar system where we incorporate culture meat into this closed loop system. We would have culture meat producing ammonia, giving it to microalgae, which would then convert it into nutrients for the cultivated meat, and then both the culture meat and the microalgae could be used as food for the crew. So there's some companies already working on these systems, mainly using algae as nutrients for culture meat. And there's a group in Japan researching circular culture systems using myoblasts and certain microalgae. Regarding the lactate, there's a similar approach that could be used. So some cells in the body can convert lactate into glucose. So we could think about a closed or co-culture system where we have culture meat cells converting glucose into lactate, and some other cells such as liver or kidney cells converting lactate into glucose and fat again into the system. In this regard, there's two companies that have patented a co-culture system using different types of mammalian cells. And there's many other considerations that we should think about. Scaffolding, do we want a scaffold that is edible or reusable? So edible, we would need a higher launch mass, or you would need to synthesize it or produce them on board, while the reusable, we would likely need to remove the cells or remove the product from the scaffold before consumption or post-processing. Regarding sensing and monitoring capabilities, we have to think that we need to measure pretty much every parameter of the bioprocess, such as biochemical metabolites, nutrients, chemical and physical parameters such as temperature, pH, and gases. But we also should be able to measure radiation. We also have to think about sterilization methods. This can be divided into heat, radiation, or chemical methods. And last but not least, the post-processing of the system. So we have to think about what kind of product we would want to have at the end, whether unstructured or structured, whether we would want to use 3D printing technologies, what kind of storage do we need? Can the product be stored at room temperature, or shall we store it at in refrigerated temperatures? And what kind of cooking methods can we use? So I'd like now to do an exercise with you. So if you think about a trip to Mars, a round trip to Mars, that could take up to 1,200 days with a crew of 4 people, each one of them weighing 82 kilograms, and this being the required animal protein intake per crew member per day. Over the 1,200 days, we would need 1.3 tons of cultivated meat for the whole crew. If you look at these values that are provided by GFI that show the amount of media that is needed per kilogram of cultivated meat, according to 3 different media formulations from low to high efficiency. And for each one of these formulations, 97% of the media is water. If we multiply these values by the total amount of meat that we need to produce, so this would be the amount of launch mass just for the inputs that we would face. So 53 tons for the low efficiency, 17 tons for the mid efficiency, and 10 tons for the high efficiency. As a comparison, here in the dashed line, we have 12 tons, which would be the amount of prepackaged food that we will need to launch with the current space food system. As 97% of the media is water, you can think that the most obvious thing is how can we recover some of this water? So if you use, for instance, water reclamation systems and water filtering systems that exist already on the IHS, you can think of recycling different percentages of this water. And if you reach 75 to 90% recycling capability of this water, you see that the amount of inputs that we need reduces significantly. So this starts being viable when compared to traditional food system. So the integration of cultivated meat systems with water reclamation systems or technologies can indeed make this cultivated system viable in space. If we now think about the bioreactor and the size of the bioreactor, we assume, let's say, a production cycle of 4 weeks, as I showed in the initial scheme. In 4 weeks, for that crew, for this crew type I established, we would need about 34 kilograms of meat over the 4 weeks. This equals to this amount of cells, and we know that per bioreactor, these are the 3 different types of bioreactors. This is the max cell density reported in literature. So to produce this amount of cells over 4 weeks, we would need this. So the internal volume of each bioreactor would be this one. So it's also pretty clear that bioreactors that achieve higher cell density will likely be preferred due to the lower volume and mass. Then microgravity, which might be one of the main obstacles. So we know that there's, for quite some years now, we know that microgravity leads to muscle loss. There's several studies on humans, but also on some animals and some in vitro studies. There's some ground studies with simulated gravity that show that myoblast cells have a reduced myogenin and myosin production, these are muscle-specific proteins, and also lower expression of myogenic markers. So most likely we would need to come up with countermeasure strategies to stimulate cells into forming muscle tissue. And the same might be similar to fat tissue, but there's not so much data on this. So this is a very simplistic concept of what the system could look like. So you would have water coming from the craft, being fed into a general media tank that is then combined with certain nutrients that are specific to culture certain types of meat or food products. We would then have some removal of metabolites and a recycling unit that could be then feed freshly produced nutrients to the media tank. You then have a post-processing unit, which could be a 3D printer, and then the storage and cooking unit. We could also think about a system where two bioreactors would be working simultaneously, one producing a certain food type, such as salmon, and another food type, such as beef, and in this way introduce also some variety in the cruise menu. So essentially, by optimizing or trying to develop these culture meat technologies for space, we would eventually reach a point where we would be using culture meat for human space exploration, but most importantly, we would be taking lessons down to Earth and applying highly optimized systems down on Earth as well. This is not only applicable to cultivated meat, but pretty much any food system in development. And indeed, by applying these three, I would say mainly these three characteristics, so reliability, stability, minimization, and integration and circularity, we would be able to come up with systems that are less dependent on resource availability. They're less dependent on climate and environmental conditions, which leads also to a more secure and safe supply chain. By having, let's say, an autonomous system, we would also be able to have decentralized and local production facilities and democratize access to certain food products independently of geographic location. So in theory, you could think about a mobile deployable unit that could be transported to remote locations to produce certain food types. So take-home message is that indeed the current space food system might not be able to provide adequate nutrition in future long-term and long-distance human space flight. And we believe that cultivated meat has some potential as a component of a future space food system, although main limitations or obstacles include launch volume and mass, recycling strategies, and cell behavior in space conditions such as ultragravity and deep space radiation. We know that bioreactors that achieve higher cell densities will likely be preferred due to volume limitations, and that recycling strategies, mainly for water, will be crucial for making cultivated meat a suitable alternative in human spaceflight. Integration with the existing life support systems will further render the system more efficient, and this includes other food production systems or water reclamation units. But ultimately, the development of and implementation of these systems in space could also lead to a great technological return down to Earth and eventually contribute to a more secure and safe global food system. In the future, we would like to study how cells or culture meat cells or cells are relevant for culture meat behave in space environmental conditions, and this is ultragravity and deep space radiation, and eventually come up or investigate potential countermeasure strategies to eliminate the effect of these two parameters. We would also like to study filtering and recycling strategies such as wastewater and recycling strategies for cell culture byproducts such as lactate and ammonia, and eventually study also the possibility for integration with other life support systems, such as I mentioned before, water reclamation units and other food production systems. I'd like to thank you for your attention. These are some links that contain some information that might be interesting for you. And this is my email in case you'd have any idea or would like to discuss the topic. I would be more than open to have a talk.